Hello, very good morning to all of you. Welcome back to the video lecture series. So dear friends, in last lecture we have we have completed the protein analysis part, and now we are going to start the characterization of proteins from this food analysis section. Okay, so the characterization of proteins, what does it mean? Characterization of proteins means which type of amino acids are present and how much they are present. When we are getting this information, then the analysis is called as characterization of proteins. This section is also related to the procedures for the different characterization of proteins given by the different group of scientists or different group of companies. Okay, we know that all the proteins are made by a fixed group of amino acids known as alpha amino acids, and these alpha amino acids are joined together by a bond known as peptide bond or a amide bond, and hence we called the proteins as polypeptides. So proteins are also known as polypeptides because in proteins a number of different alpha amino acids are joined together by a peptide bond, and hence more number of peptide bonds are present in the polypeptide, that is proteins, and hence we call the protein as a polypeptide molecules, and hence they are also called as polymers of amino acids. Hence for the nutritional value point of view, for the information point of view, we have to recognize, we have to obtain the information related to the which type of amino acids are present, how much they are present, how much more mass of that amino acids are present, of different amino acids are present in the given food sample. Okay, that information can be brought by doing amino acid analysis, hence our today's topic is amino acid analysis. Okay, so the amino acid analysis, in short I have written these amino acids as AA for time saving purpose. Amino acids are as AA. So this amino acid analysis gives us the idea of which type of amino acids and how much grams or how much milligrams or micrograms of these amino acids are present in the given protein or a food sample. Okay, so that can be identified by using a method known as amino acid analysis method. Okay, that we are going to see in detail in today's lecture. Okay, so the principle of this method is that amino acid analysis is used to quantitatively determine the total or what different type of amino acids present in the given protein or food sample. Okay, what is the composition of amino acids? What are the different types? What are the different amino acids present in the given protein sample? That we are going to determine in this procedure. Okay, in this case the principle is mainly lies or it is based on the hydrolysis of protein. We know that amide bonds can be hydrolyzed, and when amides bond can be when it is hydrolyzed, then there is a breaking of that NH CO bond known as peptide bond, and hence when proteins are hydrolyzed, okay, in a acidic condition, then we are getting a different mixture of a different amino acids. Okay, so protein sample, when it is undergone hydrolysis, it is converted to a group of a mixture of amino acids. Okay, then these amino acids, once we get after hydrolysis, then they can be separated by using mainly three separation techniques known as ion exchange chromatography, then reverse phase liquid chromatography, and last one is gas liquid chromatography. Okay, so these three separation techniques can be used to separate these amino acids from the mixture after hydrolysis. Okay, so ion exchange chromatography, reverse phase liquid chromatography, and gas liquid chromatography are used to separate the amino acids from the hydrolyzed mixture. Now let us see what is the procedure that is adapted, that is used to separate these amino acids, to hydrolyze these amino acids, to hydrolyze these protein samples into a mixture of amino acids, and the separation technique. The procedure is quite tedious and it requires a lot of time, because the separation is a very long process, and before separation the hydrolysis of proteins also takes a lot of time. Okay, but it is generally taken, or it is generally considered that 24 hours, a full day, is generally considered as a average time for the hydrolysis of proteins present in the food sample. Okay, so that's why the procedure is started in general with protein sample is first of all hydrolyzed in a constant boiling means our no doubt the solution here is made in water sample, so the constant boiling mixture will be boiled at 100 degrees Celsius. Okay, so the acidic condition is maintained, and for that we are going to use here 6 normal HCl, 6 normal hydrochloric acid is used, and this hydrolysis is done, or it is carried out for consecutive 24 hours. Okay, so for 24 hours this protein sample is hydrolyzed, and after hydrolysis we are getting mixture of amino acids. Okay, now these different amino acids undergo different different protein samples undergo different changes, and hence once they get hydrolyzed into amino acids they may undergo different changes. Okay, for example tryptophan is get completely destroyed. Okay, so that's why the time required for the complete hydrolysis of these amino acids, of these protein samples into amino acids, may vary by food to food sample. Okay, and hence duration of hydrolysis varies. Duration of hydrolysis for these foods, for these protein samples, varies as per the food changes. Okay, the some of these amino acids are get converted into their acid derivatives. For example, glutamine is converted into glutamic acid, then cysteine is converted to cystic acid, etc. Okay, then tyrosine may get longer time for hydrolysis in this 6 normal hydrochloric acid condition. Okay, under this acidic condition this tyrosine amino acid requires more time for complete hydrolysis procedure. Okay, then after hydrolysis is that completed, okay, the next step is the separation of amino acids into its components. Okay, then this separation, amino acids were separated by cation exchange chromatography. First of all, the amino acids are separated by cation exchange chromatography. Okay, in principle I have already told you that we are going to use here ion exchange chromatography, and in that ion exchange chromatography are of two types, cation exchange and anion exchange chromatography. So in this case we are going to use here cation exchange chromatography. Okay, so the amino acids are separated by, first of all, by cation exchange chromatography. Okay, and for that we are going to use here stepwise elution. We know that a column is there and this column is filled with a cation exchange reagent. This column is filled with a cation exchange reagent and it is loaded with a mixture of amino acids. A mixture of amino acids is loaded along with the cation exchange resin. Okay, and this is the elution solvent, the solvent used or the mobile phase used for this elution process is a solution with increasing pH, solutions with increasing pH and increasing ionic strength, increasing pH and increasing ionic strength. So this is the criteria used for the separation of amino acids by using cation exchange chromatography. So as pH is increasing, as ionic strength of that solution is increasing, the separation occurs, separation of amino acids occurs from this mixture. Okay, so this is the main basic theme or main basic idea behind this cation exchange chromatography. Okay, then after once the elution, once we are getting the elution, that eluate that is obtained from the elution process from this cation exchange chromatography, then these amino acids are get separated here, amino acids are get separated here by using cation exchange chromatography, and we are having now a fractions, we are having a fractions of amino acids having different, means they are separated. If a sample is containing 10 amino acids, if a food sample is having a protein and that protein is having 10 amino acids, that protein is made up of 10 amino acids, then we are having 10 aliquots or 10 fractions of different 10 amino acids. Okay, so here by using cation exchange chromatography we have separated the amino acids present in that food sample. Okay, then the next step is post derivatization with ninhydrin. Post means after. Okay, so post derivatization means after column chromatography, after ion exchange chromatography, we are going to do a derivatization of these amino acids, derivatization of amino acids with ninhydrin. So ninhydrin is a reagent. The structure of this ninhydrin is like this. It is, this structure is the hydrolyzed form of ninhydrin. Okay, that ninhydrin is also written like this. The structure of ninhydrin is also written like that. It is a trione. Okay, so 1, 2 and 3 carbonyl groups are present adjacent to this, adjacent to one another. Okay, aromatic ring, benzene ring is there, and it is fused with 5-membered ring. Okay, and there are 3 carbonyl groups CO, CO, CO are there present in this compound. This is the structure of ninhydrin, and when water molecules get react with this ninhydrin, okay, we call it as a hydrolyzed form of ninhydrin. This is the hydrolyzed form, and when this ninhydrin is reacting with our amino acid which is separated with our by using cation exchange chromatography, these amino acids contain NH2 group, a free NH2 group is there, and this NH2 group is responsible for the reaction with this ninhydrin molecule, and hence this ninhydrin reacts with this amino acid separately. Okay, because we have separated forms of all amino acids here, and we are derivatizing each and every amino acids with this ninhydrin molecule, ninhydrin reagent, and the reaction, general reaction I have written here, this ninhydrin reacts with these amino acids containing NH2 group. Okay, and such colored complex, the structure of this complex is written like that. This R is taken general, means R is any side chain amino acid, R is side chain of any amino acids. Okay, so this type of structure moiety is generally formed, and this is giving a colored complex, this is giving a colored complex. Okay, so that's why sometimes, in most of the times, blue or purple color complex is generally formed with this ninhydrin reagent, and that is detected using UV visible or fluorescent detector, UV visible or fluorescent detector. Okay, so that's why this is the general reaction between this ninhydrin reagent and an amino acid. Okay, so that's why this technique is known as post derivatization with ninhydrin. Post derivatization means after cation exchange chromatography, when we have separate samples of amino acids, then each amino acid is then reacted with the ninhydrin molecule, ninhydrin reagent, and then each ninhydrin reagent is giving a separate colored complex with separate amino acids. Okay, then they are treated as this is a colored complex, then we are going to use absorbance spectroscopy. Okay, so means by using spectrophotometric analysis we are going to use the concentration of each amino acids by using a chromatogram. Okay, so here a chromatogram for a general chromatogram is shown here for the separation of amino acids by using this technique. Okay, so here absorbance is plotted on the y-axis against time on x-axis. Okay, so here the number of peaks can be seen here in this chromatogram. Each peak is responsible for each different amino acid. For example, we can give a number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 up to 20, 21, etc. Okay, so all these peaks are responsible for a different amino acid. Okay, so when the internal standard for this chromatogram is generally used, it is norleucine. It is generally norleucine. Norleucine is also an amino acid, but this norleucine is not found in the food samples. Okay, so that's why it is generally used as an internal standard in this chromatography. Okay, then calculation part. So the calculation part of this it is generally done by spiking the sample with known quantity of internal standard. By spiking the sample, by doing measurement and area under the peak, by measuring the height and area under the peak for each peak in this chromatogram, we call this process as a spiking process, and the sample with known concentration of known quantity of internal standard, all other peaks are spiked or they can be estimated by using a inbuilt software in the given liquid chromatography. Okay, then the results of these are expressed as mole percentage. The results of these chromatogram, either reverse phase liquid chromatography or gas liquid chromatography, are generally expressed as mole percentage. So how to obtain mole, that we know at this stage, but how to obtain mole percentage, that we are going to see here. First of all, we will see how to obtain how much amount of mole present of each amino acid in the given sample. Okay, so for that, calculate moles of each amino acid by taking a ratio of mass divided by molecular weight. So molecular weight of each amino acid we can know by doing calculation or by literature. Okay, so mass of amino acid can be obtained from this spectroscopy data from this chromatogram. Okay, then once these moles of amino acids of each different amino acid has been calculated, then do a sum of moles of all amino acids. So it will give you total number of moles present in that amino acid mixture. Okay, it will give you total number of moles. Okay, then divide these moles, divide mole of each amino acid, that we have obtained in the first step, mole of each amino acid divided by the total number of moles. Mole of each amino acid divided by total number of moles, you will get the mole fraction. Okay, and then multiply this value by 100. Multiply this value which is obtained in the third step by 100. You will get mole percentage. Okay, so this is the stepwise calculation to obtain the mole percentage of amino acids present in the given protein or food sample. Okay, then the last point is applications. So what are the applications? It is very general or it is very routine that what is the use of this amino acid analysis. This amino acid analysis is used to determine amino acid composition of a protein, no doubt. As the title indicates, this gives the information about total composition of amino acids, which different types of amino acids, how much they are quantitatively present in the given food sample. This information can be brought from this amino acid analysis. Okay, then to determine quantities of essential amino acids, quantities of essential amino acids is evaluated by using this analysis. Then to evaluate protein quality, so how much protein is present and how much different amino acids are present, okay, that can be obtained by using this amino acid analysis. Then this information is also useful for the nutritional point of view. Okay, so we already known that for giving a label on the different food products, nutritional value is essential, and to obtain that nutritional value, this amino acid analysis is also useful. Okay, so these are the different applications of these amino acid analysis. Okay, so this is the general information and the content of this amino acid analysis. Okay, so this is the first lecture related to the characterization of proteins. The chapter is same, that is chapter number 6. Okay, but it has two parts. The first section is related to the protein analysis, and second section is related to the characterization of proteins. Okay, so I think you got it. Thanks for watching this video. Have a nice day.